So we'll continue with adaptive immunity. So adaptive immunity is a specific immunity that has memory. It is an immune response that has to do with the lymphocytes and their products, right? It has a longer response time than the innate immunity, but it's way stronger. It's more thorough. We call it the third line of defense. However, we don't abandon the first two. So while this is going on, we're still keeping our membranes intact, and we're still having complement and interferon and phagocytes and natural killer cells working, and fever and inflammation. Like, all of that is still happening. There's two branches of adaptive immunity, which is also called specific immunity. There's the cell-mediated immunity, where cells are actually interacting with other cells. And this is T-lymphocytes that do this. And then there's, humoral immunity. Humor is an old word for fluids, for body fluids. So humoral immunity has to do with non-living extracellular things, proteins really, that are put out into the fluids. And so this is B lymphocytes, plasma cells, and antibodies. So it's often called antibody-mediated immunity. So cell-mediated and antibody-mediated or humoral immunity. So we'll start with antigens. What are antigens? Antigens are the things that evoke the immune response. They are the features of the pathogen that actually evoke the response. Usually it's a protein or polysaccharide or a glucolipid or something like that. They tend to be large organic molecules. So it can be part of the protein capsule of the viruses, proteins in the cell wall of bacteria or fungi. It can be toxins that are produced by bacteria. It can be just any abnormal proteins that are around. They are antigen. They generate the anti. They generate the response, as it were. So there are foreign antigens that our body will attack and deal with, and there are self-antigens that our body uses to recognize our own cells. That if I was to give you tissue, my self-antigens would be foreign antigens to you, and your immune system will attack it. The immune system can usually distinguish. However, if they can't distinguish, that's where autoimmune disorders come from, like rheumatoid arthritis and diabetes mellitus type 1 and things like that. So when we look at the antigen, it's what is it about the antigen that actually evokes the response. And we call this the antigenic determinant. And each determinant has a special shape, and the antibodies, or the receptors on the immune cells, have a corresponding shape. The analogy that I use is, what do you find attractive in another person? Do you like tall or short? Do you like dark complexion? Do you like light complexion? Do you like brown eyes? Do you like blue eyes? Do you like that they have a good job and lots of money, or does that not matter? A nice car, etc. Come from a good family. All of these things are determinants that that evoke a response, a physiological response in you. And the more of those determinants that an individual has, the stronger the response there will be to that person. So it's the same sort of thing. The more antigenic determinants that are present on a pathogen, the stronger the immune response against that pathogen will be. And having this response is called immunogenicity, the ability to trigger this response. So an immunogen is really the antigen that has immunogenicity. Now, there are some things that will react, and they're kind of half antigens. We call them haptens. They're not large enough to evoke the response. They don't have immunogenicity on their own. They become immunogenic when they combine with proteins that, or a carrier molecule that is in the body already. For instance, the oils in poison ivy need to bind and make a complex with proteins that the body produces in order to become an antigen and have a reaction to. Animal dander and things like that often fall into this. So this is really where allergies come into play, is this idea of haptens. So the lymphocytes have these receptor complexes, and I don't really care about TCR and BCR, but it's really the T are on the T and the B are on the B cells. But each one, the point is that each complex binds with one specific antigen, and only one. It ignores all the other antigens. There is a different T lymphocyte or B lymphocyte for a different antigen. And so we have to have thousands and thousands and thousands of different B and T lymphocytes. So the lymphocytes become activated when they meet their antigen, the antigen that they have a receptor to. Now, there's two ways that this happens. When the antigen is on the pathogen and it meets a B lymphocyte, the B lymphocyte becomes activated, because the B lymphocyte's job is to produce antibodies, and antibodies work against the pathogens in the interstitial fluid. So they're going to be activated by pathogens in the interstitial fluid. Now, T lymphocyte's job is to kill a cell, on your own body cell, that has something wrong with it. So like a virus has gone inside it. When the virus goes inside it, the cell stops making the correct proteins and makes viral proteins instead. So that means its surface proteins start to have viral proteins in it. So the antigens are processed and they become part of the coat of the host cell. And since that's what the T lymphocyte's job is to find the cells that have that, it has to be activated by a processed antigen that has been processed and presented to it by something called an antigen presenting cell. So a macrophage will eat a pathogen, process the antigen, place it on its surface, and then present that antigen as a processed antigen to a T lymphocyte and activate that. There's a bunch of different types of T lymphocytes, but the two fundamental ones that we care about are the helper Ts and the killer Ts. Cytotoxic, kills cells. There's memory T cells, there's regulatory T cells, there's suppressor T cells, there's a number of different ones, but we're going to concentrate on helper Ts and cytotoxic Ts, killer Ts. Okay, so where do these lymphocytes come from? It's the second most common white blood cell. Never let monkeys eat bananas. Lymphocytes. They are formed in the bone marrow. And they're formed as kind of a generic lymphocyte. And then they get the receptors to this specific antigen, and they become immunocompetent by getting the receptor that they need. One population of them get that receptor right in the bone marrow where they're made, and those are called the B lymphocytes. Another group, another population, migrate to the thymus, and they become immunocompetent in the thymus, so that's why they're called T lymphocytes. Now, both of them, when they become activated, the B lymphocytes by a free antigen on a pathogen, the T lymphocytes by a processed antigen and an antigen presenting cell, what they do is, as soon as they're activated, they start making clones of themselves. They start reproducing, they replicate, and they form a whole bunch of identical lymphocytes with the identical receptors on them. They don't have to go to the thymus or the bone marrow to get it. They just become clones, so that you basically build an army of the lymphocytes that are designed to go after that antigen. Because the whole point of the adaptive immunity is to eliminate the antigens. But this takes a little bit of time. So these activated lymphocytes that have cloned, they end up migrating to the site of infection. And they work there, especially the T lymphocytes. They have to go right to where the problem is because it's cell-to-cell interaction. The B lymphocytes can stay in, and they make antibodies, and they go into circulation, and they end up working everywhere. But when the B lymphocytes clone, they make huge numbers of copies of themselves. Some of the clones start to grow a bunch of endoplasmic reticulum, which is a factory for making proteins, read antibodies. As they grow this endoplasmic reticulum, they have changed. And so now we call them plasma cells, as in endoplasmic reticulum. And the plasma cells are the ones that make the antibodies. And then the antibodies are transported to the infection site through the blood and through lymph. So that's the formation. And this is activation. Okay, so the effector cells, they are helper T cells, cytotoxic T cells, and plasma cells. We already just said plasma cells produce antibodies. We'll get into what they do in a minute. Cytotoxic T cells destroy our own body cells that have something wrong with them. And the helper T cells coordinate the action of both of these things. They release cytokines, and they regulate cells in the adaptive and in the innate immunity. The helper cells will make natural killer cells become more active. They'll make phagocytes become more active. They're involved with chemotaxis. But they also activate and stimulate the killer Ts. They do what's called co-stimulating. And again, we'll use an analogy. If you've ever watched Flashpoint, you have a SWAT team. The sniper goes up on the roof, picks out the bad guy in the scope, has a shot, can kill the bad guy. But he doesn't. He calls the captain in the Winnebago that's coordinating all the responses and says, okay, I have the target. Do I take the shot? And then the guy in the Winnebago says, yes, take the shot. And the bad guy gets killed. the T lymphocytes work exactly the same way. The killer Ts do the killing. The helper Ts give permission for the killing to happen. Because we're killing our own body cells, that can be expensive to us, so we don't want to do it haphazardly. So the helper T's job is to identify that the bad guy is there and then be the second stimulant. So the bad guy stimulates, like the processed antigen stimulates the killer T to target that cell, and then it also stimulates the helper T, and then the helper T gives permission to the killer T to do the killing. So you need that co-stimulation to happen. Now, the helper T can also activate B cells, the correct B cells, to clone and produce effector cells, plasma cells, to make antibodies. And the helper Ts can all, like I said, do other things with the innate immunity. It just enhances everything. Now, when these clones happen, not all of them become active. A population of them, a certain percentage of them, stay as what are called memory cells. And their job is to be prepared to meet the same pathogen or the same antigen again sometime in the future. Chances are good that if you've run into a pathogen, you're going to run into it again. So you better be prepared. And the fact that there's so many more memory cells after the first time you've gone through this, the second time you go through it, it's much faster and much stronger. So, so these are the things that the helper Ts do. Help activate Bs, activate cytotoxics, co-stimulate them, and stimulates activity of the phagocytes and the natural killer cells, etc. So the killer Ts, after they've been activated, they make a bunch of memory cells, and then certain of them become effector cells, and they go to the site of the infection. They make physical contact with the foreign cell or the unhealthy cell. They get co-stimulated by the helper T, and they release perforin that just eats holes in the target cell membrane and kills the cells. When that cell is killed, then the viruses or in the cell come out into the interstitium where they get eaten by macrophages. When you activate the B lymphocytes, you get the clone, you get a bunch of memory cells, but most of them become plasma cells, and the plasma cells pump out antibodies by the millions for about 5 days. And the antibodies circulate and end up being everywhere in the body, and they help deal with the antigens, with the foreign. These are called immunoglobulins, IGs. Now there's 5 different types, and it's MAG, IgM, IgA, IgD, IgG, and IgEs. They're Y-shaped things. We don't really care, but they are specific to specific antigens. So what do? They neutralize. They basically swarm the pathogen. So say it's a bacteria. The bacteria will get so many antibodies stuck to it at all of its antigens, all of the proteins on its surface, that the thing can't swim, it can't interact, it can't do anything. So it gets neutralized. And it can't get away from the macrophage that's going to eat it. Another thing that antibodies do, the IgM antibodies, they basically join pathogens together. They handcuff them together like a chain gang, so they can't run away, and makes it so that the macrophages can eat them. So we call that agglutination, gluing them together, clumping. Another thing that antibodies do, they get so many of them on the pathogen that the pathogen, especially the viruses, fall out of solution and they precipitate and therefore don't circulate as well, so the macrophages can eat them. So neutralization, agglutination, precipitation. And this is what pictures what they look like. They also fix complement. So when we were talking about complement, inserting the membrane attack complex to lyse the cell, the bacteria, how do we know what cells to stick complement in? Well, we stick it in the places where antibodies have attached to antigens on that cell membrane. So the antibody attaches to the pathogen, which then directs the complement proteins to go there. And it creates this opsonization, the handle that the macrophage can grab hold of the pathogen so that it doesn't get away. So it enhances phagocytosis. Some of these antibodies will actually activate NK cells, because what they will do is they will tell the NK cell that there's something wrong with that cell, and so that cell will get attacked. But the most important things are neutralization, agglutination, precipitation, and activate complement. Now, all of these things take time. There's a lag time. The first time you encounter a pathogen with these antigens, it might take 3 or 4 days before you are ready, like that you have this response. In COVID, it seems that it's taking up to 2 weeks, because you're asymptomatic, although you're filled with the virus, and the symptoms are caused by this fight. And so the inflammation and all of the things that are going on, it takes a while for this response to happen. So there's a lag time between the first exposure and what's going on. But when it gets activated, you get all kinds of memory cells against that specific antigen. So the second time you encounter it, you have all these memory cells, and it doesn't take as long to have a response. Because more memory cells make more contact with the antigen more rapidly, and you have this powerful response. And that's kind of the point of a vaccine, is to develop this memory. By exposing to the antigen rather than the pathogen, you will evoke this response, make the memory cells, and then have the memory cells for if you encounter the pathogen for real. So that's the whole point of vaccination. And we're not going to talk about HIV and AIDS with this. Now, there's basically 4 ways to acquire this immunity. There are 2 ways that are active, or 2 ways that are natural, and 2 ways that are artificial. The natural ways of getting it are to be exposed to the pathogen. So you get sick, you're exposed to the pathogen. Like when I was a kid, I got the measles. I got the German measles. I got the mumps. I have immunity to those 3 diseases because I had them. And now I have been actively making the memory cells and the antibodies against them. You guys probably got the MMR vaccine, which is exposing you to the antigens, not the pathogens. So you artificially got the antigens. You got an injection from the doctor, so you were vaccinated against them. And now you have protection actively because you have made the memory cells, and you have active. Both of those are active because you've had the immune response. First one natural, the second one artificial. You can also have passive. And passive is when somebody else makes the antibodies for you. So the natural way of this happening is that your mother, while you're in the womb, gets exposed to a pathogen, makes antibodies against that pathogen. Those antibodies cross the placenta, end up in the baby's blood, and then when the baby is born, that baby has a couple of months of protection against the pathogens that are in the environment that it's born into. It's a natural passive. The baby doesn't have its own immune response. It has protection from the antibodies made by the mother. The mother can also pass that through breast milk. Antibodies are in breast milk, and that helps protect the baby as well. That's a natural way of having passive immunity. There's also an artificial way of having passive immunity, and this is gamma globulin shots. This is the classic one is snake venom. If you get bit by a cobra, you don't have the time to have an immune response against that cobra venom. You don't have days. So what they do is they take the cobra, they milk the venom out of it, they give little bits of the venom to a large animal who will not die, but will make antibodies against that. Then they harvest the blood from that horse or whatever. They centrifuge out and they extract the antibodies from that animal's blood, and now they can use that as a basis to clone it, to make monoclonal antibodies. And then they put it in a vial, and you get bit by a snake, you go to the hospital, and they inject the antibodies into you, and so the antibodies neutralize and precipitate and agglutinate the venom. But they're not your antibodies. They were made by, and it didn't come naturally. They came artificially via an injection. So that's it for adaptive immunity.